Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by an independent educational grant from Novo Nordisk AS.
This program is presented by Medscape Education Global.
Welcome to this program titled Unveiling the Bigger Picture of Alzheimer's Disease from New Disease Mechanisms to Biomarkers. My name is Philip Scheltens, Professor Emeritus at the Amsterdam University Medical Center in the Netherlands, and I'm joined by Professor Frank Jessen, who is Professor of Psychiatry and Director of the Department of Psychiatry at the University of Cologne in Germany, and Professor Hendrik Zetterberg, Professor of Neurochemistry and Chief Physician at the Institute of Neuroscience and Physiology at the Sahlgrenska Academy at the University of Gothenburg and Mölndal, Sweden. Welcome to this exciting program. So why are we here today? Well, we are here because so many things have happened in the field. We have new discoveries, renewed interest in new targets and therapies for Alzheimer's disease, and especially, I would say, the enormous adventure we have taken over the last 10, 20 years in biomarkers, and you will hear about this during this program. That's exactly a key element, using biomarkers to detect early signs of Alzheimer's disease, and biomarkers to also be used to detect therapy effects and also monitoring the effect of therapy over time. So we will discuss the role of these biomarkers in diagnosis, but especially also in developing therapies, and we'll focus also on neuroinflammation, particularly the GLP-1 receptor agonists. First, Frank, I will ask you to give us some background on the pathophysiology of Alzheimer's disease. How is it all interconnected?
Yes, of course. So the two key pathological features of Alzheimer's disease are amyloid plaques, which are in the extracellular space, and intraneuron fibrils called neurofibrillary tangles composed of aggregated tau protein. And these are the hallmarks of the disease. The idea is that amyloid plaques are first, and they stimulate tau aggregation within the neurons, which then leads to neurodegeneration. One important finding from the last years is that there is another key player in this game, and this is neuroinflammation. By the amyloid deposition, most likely microglia, so the immune system of the brain, is activated, and there's also a release of cytokines, so inflammatory mediators. And we think today that these inflammatory activations are actually also detrimental for the brain cells and add to the toxicity of amyloid and tau. Looking a little bit more deeply into the neuroinflammatory part, so what happens is that these amyloid species and also probably tau in an interplay with certain risk factors, for example, genetic background and age and other environmental factors, stimulate the brain immune system. We know, for example, from genetics that genetic variants which are associated with the inflammatory pathway are actually also risk factors for Alzheimer's disease. So that is quite a strong piece of evidence. This all leads to also other phenomena, like, for example, disruption of the blood-brain barrier and other toxic processes. And this all accelerates neurodegeneration. Another piece of this puzzle is activated oxygen species, or ROS. We know today that ROSs are released by mitochondria and also by the activated immune system. And the problem is that ROS and also nitrogen species are also neurotoxic and are associated with neurodegeneration. So this is kind of like a vicious circle between amyloid and tau, and then the activation of microglia, the release of oxidant species and nitrogen species, which all accelerate the neurodegenerative process.
I think it has become clear that onset of amyloid pathology could at least potentially relate to cerebrovascular health and clearance mechanism. This protein is produced by all of us a lot, and aggregation is concentration dependent. And if the brain is not flushed regularly by brain fluids, and if the cerebrovasculature can't deliver oxygen and maintain a good health of the brain cells, then you are at increased risk of getting amyloid pathologies. We can measure it using biomarkers, both amyloid PET, cerebrospinal fluid markers, and now also blood tests. And we know that this pathology appears like 15, 20 years before dementia symptoms are evident. And then this pathology is followed by synaptic dysfunction and neurodegeneration that eventually will give you clear evidence of tau pathophysiology, and then neuronal loss, and then the symptoms come. So there is an interesting link there between the cerebrovasculature, amyloid, neurodegeneration, oxidative stress, inflammation, all those.
So there is no Alzheimer's disease without amyloid. Would you say that?
Yeah. Per definition, it has to be like that.
Yes.
So neuropathologically, you can't give an Alzheimer's diagnosis to a person who is amyloid negative. But then, of course, we have amnestic phenotypes with memory dysfunction.
Yeah, the real amnestic dysfunction. So you have shown us the trajectory, but how are we going to measure this trajectory? I mean, where do the biomarkers come in?
Yeah. So this is where the field has developed enormously during the past 20 years, I would say. So we can measure the amyloid pathology with cerebrospinal fluid tests of beta amyloid peptides, and the best and clinically approved and FDA approved and everything biomarker now is the beta 42/40 ratio. And that really also relates very beautifully to amyloid PET positivity. So if you're amyloid PET positive, you are very likely to also have a low CSF beta 42/40 ratio. This can also be measured in blood now, and that's a new thing. But as soon as amyloid plaques appear in the brain, you get tau phosphorylation and secretion. And those phosphorylated forms of tau can be measured in cerebrospinal fluid and in blood. And from my point of view, they work almost like amyloid pathology reporters.
And some drugs are targeting neuroinflammation, but can we also measure it? I mean, is this also something that we can detect? Frank?
Yes, we actually do have biomarkers for neuroinflammation. We do have markers, for example, for the activated astrocytes, which is also a component of the brain immune system. So microglia and astrocytes are activated in the immune response. And we have one protein which is called YKL40 in the CSF. And even more prominent is a biomarker which has really been studied a lot over the last years in Alzheimer's disease, and that is glial fibrillary acid protein, or easier GFAP. And other markers, for example, for activated microglia is TREM2. And we also have one marker for neuronal degeneration, and that is neurofilament light, or NFL. So I would like to pass over to Henrik to tell us more about these markers of neuroinflammation.
And then we can measure in cerebrospinal fluid proteins that are released from microglia that are activated by plaques. And in the literature, the most studied protein is called soluble TREM2. And then it looks like, in longitudinal biomarker-based studies, when people become amyloid positive, they get an increase in these microglial activation markers.
Interesting.
And then in parallel with this, what happens is that the astrocytes also react in an inflammation-like manner, getting swollen, releasing a protein called glial fibrillary acidic protein. And then both microglia and to some extent astrocytes can also shed some interleukins and cytokines. And this is possible to measure in cerebrospinal fluid. It's much harder to pinpoint this in blood with one exception, and that's this glial fibrillary acidic protein.
GFAP. Yeah.
It's surprisingly good. So when amyloid plaques appear, GFAP levels in blood increase in parallel with the phospho-tau increase.
But you do say, I mean, that's good for the audience to realize, this only happens when amyloid pathology already has started.
Exactly.
It's not the reverse.
I've thought about it. We don't have a single biomarker that helps us predict amyloid plaque pathology onset. Except for some genetic markers, of course. That leads us
to the next question, the genome-wide association studies. Here you see on this particular slide, you see on the left-hand side, you see a few genes that actually are totally on the top that are the really causative genes for Alzheimer's disease. PS1, PS2, APP, actually also a SORL1. If you go more to the right, we're talking more about risk-associated genes. So for instance, ApoE4 is there, but also TREM2, that Henrik also discussed, is an important gene that actually increases your risk for Alzheimer's disease, but doesn't cause the disease. So we have this GWAS studies, huge amount of patients, hundreds and thousands all over the world, have shown that there are so many genes involved. So ApoE and TREM2 are important ones to mention again, because also they relate to the microglial response. So here again, neuroinflammation comes around the corner, as I would say. There is much ongoing in this area. We have talked about this, but what have we learned from older biomarkers, so the first generation of biomarkers in terms of inflammation, and how do we use this actually in current practice?
I think this is a very important question, because when I teach medical students, in clinical neurochemistry, we measure neuroinflammation in neuroinflammatory conditions, and that's sort of, per definition, something which is really linked to the immune system and inflammation. And then I'm thinking about biomarkers like cerebrospinal fluid cell counts that many among you listening will have experience with. And then oligoclonal bands, for example, of IgG or IgM, and also IgA. And blood-brain barrier dysfunction. And if you have a very active neuroinflammatory disease or neuroinfectious disease, then those markers become really abnormal. This is not the case in Alzheimer's disease.
No,
so I even have a slide for medical students that Alzheimer's disease is not a neuroinflammatory disease, but that goes against a lot of the new data that that has appeared thanks to the new biomarkers. So if we think about the biological markers for Alzheimer's, we have cerebrospinal fluid and plasma beta markers and tau markers, core features of the pathology. For sure. But then we also have neurofilament light as a neurodegeneration marker. But in addition, we see that proteins that are linked to the innate immune system, microglia, astrocytes, they can also be deregulated, indicating an inflammatory component.
So if you look at what is here on the slide, you have a whole array of fluid biomarkers.
Exactly.
There's so much that we can
measure
oxidative
now. Stress, definitely. 10, 15 years ago, there was a lot of hope for these isoprostanes and as biomarkers. But they turn out to be relatively general also to neurodegenerative diseases.
Not specific for Alzheimer's.
Okay. But that's also good, of course.
True, of course. Because you can measure perhaps progression over time of neurodegenerative.
And the inflammatory markers that we know the most about, which then are not the crude traditional inflammation markers, but more related to Alzheimer's pathophysiology, include YKL-40, GFAP, and soluble TREM2. I also think this is interesting because these markers link, to some extent at least, the pathophysiological process of Alzheimer's disease to neurovascular dysfunction.
Now, Frank, can you give us a brief overview of some of the data on some of these biomarkers?
Yes, sure. There are a number of studies, actually quite a rapidly increasing number of studies, showing that GFAP, so the marker of astrocytic activation within the immune response, is increased in already MCI patients, and even more so in patients with dementia, in comparison to healthy individuals. And the same is true for NFL, the marker of neurodegeneration. Both GFAP and NFL can be measured in blood, by the way, and blood NFL is also increased in many neurodegenerative diseases, including Alzheimer's disease. Not so much at the MCI stage yet, but at least at the dementia stage. And it's very clinically also very interesting to know and to understand that GFAP levels actually also predict cognitive decline. So in a person with only mild symptoms, for example, mild cognitive impairment, studies show that high GFAP levels predict the speed of decline. So the higher the GFAP, the faster the speed of cognitive decline longitudinally. The same is in a way true for NFL, not so much. The prediction of NFL for future cognitive decline is a little bit less strong than for GFAP. And NFL also increases a little bit later in the disease, but it is a very good marker for disease progression. So NFL is actually a candidate to track neurodegeneration over time.
NFL is also in FTD
It's
and
increased.
ALS,
Oh.
and you see it in all other diseases as well?
Exactly.
Yeah.
And you also see it in cerebrovascular disease. And,
Yeah. So NFL is not to be used diagnostically, you would
No,
say?
I would say that, it's my laboratory physician biased perspective here, but NFL is diagnostic for neuroaxonal degeneration.
Yes.
That's
Whatever the cause is.
whatever the cause
Yes,
is, exactly. So it's not
But
diagnostic
GFAP, is that, how important is GFAP?
Yeah,
Diagnostically or prognostically?
I think GFAP could have a great value when we evaluate treatments,
Yes.
especially treatments that are linked to inflammation or inhibiting the inflammatory component of inflammation and also cerebrovascular health.
Yeah.
GFAP is relatively short-lived, and if a treatment does something to something good to these processes, then GFAP levels should decline.
Yeah. So perhaps GFAP of NFL could actually be of
GFAP
use?
and NFL, and then there are convincing data, I think, that the level of phospho-tau increase actually predict a little bit the speed of the disease also. And if you follow people with amyloid pathology and mixed forms of pathologies, the mixed pathologies that can give you Alzheimer phenotypes, then NFL levels increase over time with a slope which is higher than what you see in people without these pathologies. These markers are also, they change in response to successful treatment.
Especially NFL.
And that we have learned now from the anti-amyloid antibody trials that if you treat with anti-amyloid antibodies, phospho-tau levels will go down a bit.
And NFL?
NFL, unfortunately, it's a slow marker. I would have loved it to decrease actually. That would have been such a clear.
But it doesn't happen.
It doesn't really happen. No. But I think it might happen if we continue.
Yeah, of course.
It's a little bit longer. It's a slow marker.
So that's research. Back to the clinical practice. Can we use them in the clinical practice? From a personal point of view, yes. We've been doing this for, I think, 10, 15 years already. But actually, interesting, in Amsterdam, we are now doing parallel blood-based biomarkers and the CSF biomarkers at the same patient at the same time, comparing it to get a feeling of how these p-tau or 181 or p-tau 217 marker actually work in clinical practice. I can tell you it's not one-to-one yet.
That's our experience too, actually. So that's why I think it's important to use these blood biomarkers clinically in a careful manner. And there are suggestions now on perhaps avoid to make statements on biomarker concentrations that are close to the cut point, and instead look at clearly abnormal, clearly normal,
and you have a gray area.
And then you can we are in the wonderful situation that we can do a CSF on those.
So that's on the diagnostic level, and now we have sort of the therapeutic landscape moves from anti-amyloid, anti-tau to anti-inflammatory therapies and oxidative stress. Let's see whether we can use these biomarkers also for that purpose. And this is interesting, then you have to review actually the AD drug development pipeline. This is the Jeff Cummings Wheel of Fortune, we always call it. And these are the different stages, stage 1, phase 2, phase 3. We have disease-modifying therapies, biologics, we have disease-modifying therapies, others, and we have symptomatic treatments. And all the yellow ones actually, if you can see it, they indicate the inflammation/immunitive sort of therapies directed at the inflammatory system. The good thing about this figure is that there's a lot of action. There's a lot of sort of in every phase, A lot of them are disease modifying, only 1/3 is actually symptomatic, and a part of those are all targeted the immune system. So that's an important message, I think, to take home. We see this also on the mode of action again. This is another figure from Jeff Cummings. You see that oxidative stress and inflammation are really important targets. And if you look the number of agents that are being tested in phase 1, phase 2, phase 3 on this particular non-amyloid, non-tau target, which is really, really important. So people have viewed the Alzheimer field as, yes, it's very amyloid-centered, it's also tau-centered, but many other things are happening there, and many other things are being targeted at the moment. So I think that's good news.
I think that's good too. It feels good. And I also think we already now can say that it's clear also from the anti-amyloid treatments that they don't solve the whole. We need to look into this.
Well, I think that's a good point actually, because you see the slowing of the slopes, you see the changing of the slopes, but it's actually not sort of upward. People are not improving, so people are still deteriorating. So other processes are ongoing still. Yeah. So as one of these repurposed therapies, actually GLP-1 receptor agonist, I mean, I don't think anybody else that looks at this stick program has not heard yet of the GLP-1 receptor, because a lot of people use it for several instances, but it's also being studied for Alzheimer's disease. Frank, can you get us started? What are these agents exactly?
Yeah, so GLP-1 in itself is a hormone which is in the body. It is released in the gut after food intake, and its role is to actually increase insulin in a glucose-dependent manner. So it regulates, it's a cofactor with insulin to regulate the blood sugar after eating. The drug class of GLP receptor agonists are used in clinical practice for the treatment of diabetes and also for the treatment of weight loss. And interesting about this drug class is that it does not only regulate insulin and glucose, but it has a wide variety actually of effects on other parts of the body. And there's also evidence that it has neuroprotective effects or effects on neuroinflammation. We know this from a number of studies in mice mainly, where it has been shown that GLP-1 receptor agonists have an effect on microglial activation. So they suppress the activation of microglia to certain stimuli, which is considered to be protective or decrease the toxic effect of neuroinflammation. So it's neuroprotective in the context of neuroinflammation. It also has effects on the astrocytes, and also it reduces the oxidative stress levels or the concentration of reactive oxidative species. And this leads to overall anti-inflammatory effects also in very relevant brain regions, for example, in the hippocampus. In addition to directly impacting on microglia and astrocytes, as mentioned before, there is also a direct effect on the mitochondrial production of reactive oxygen species, so a protective effect by this drug class. And one should also mention that these effects are not specifically restricted to GLP-1 receptor agonists, but other types of diabetes drugs, for example, SGLT2 inhibitors. There's also evidence for similar effect of this drug class, so anti-inflammatory effects and antioxidative effects.
Hendrik, back to you. After you heard about Frank, GLP-1 receptor agonist, cognition, clinical, real-world studies in diabetes. What can you tell us about this?
Yeah, I think it's, to me, this was a little bit of a surprise, a positive surprise, of course, and it's related to observational trials or intervention trials in diabetes where cognition was monitored also.
Yeah.
And this has been in type 2 diabetes, where it has been clear in these studies that have been large and relatively long that the incidence of dementia was lower in the active
And
arm.
now I have a tricky question for you. Do you think that is because of using the GLP-1 receptor agonist just improves your diabetic sort of status, or is it the inflammatory part of it?
No, I would say it's not known. It's really. So I think I see test tubes from patients and I don't see the patients. But when I listen to colleagues, I mean, in the memory clinic, if there is a deregulated diabetes, it's really, you really try to take care of that. And then cognition can improve by just that intervention.
Yes, exactly.
And I think that's important. There is still, if we move on to look at some of the other trials, there is more evidence supporting that there is an effect if you use GLP-1 receptor agonists in type 2 diabetes on cognition. And it seems to be relatively biological. At least there are indications in people who are particularly high in NFL and the phosphatide measure that they are the ones that really benefit the most. And then here, in this study, there has been stabilization of the NFL levels. And I think that's really...
Again, that's illustrating the beauty that we can do this nowadays in plasma.
Yeah, and that's why I think also it's so much more interesting to... Looking at the clinical trials that are ongoing now in Alzheimer's disease, they are much more interpretable to the outside. We can look at these markers and see what's happening. So I would say that the jury is a bit out regarding the mechanisms.
Regarding the exact mechanism.
And it will be important to do studies to examine this in greater detail. And I think Frank might actually have something to add.
We'll go back to Frank again. The studies that are ongoing in AD with the GLP-1 receptor agonist, can you tell us a bit about those?
Yes, there's actually a very exciting data pool out there, primarily from studies in patients with diabetes. So there are large randomized controlled clinical trials on individuals with diabetes treated with GLP-1 receptor agonists. And secondary outcomes of these studies were, in some cases, cognition. And it has been shown in these studies that those patients with diabetes who receive these drugs, they actually have a reduced risk of cognitive decline in comparison to those who were receiving placebo. There are other studies which started in Alzheimer's disease patients with this drug class. First studies showed, for example, positive effects on glucose metabolism. In these studies with Alzheimer's patients, these GLP-1 receptor agonists led to a better glucose uptake, indicating more activity of the neurons. And these and other findings have led to actually a small number of clinical trials in mild Alzheimer's disease, early Alzheimer's disease with this drug class, looking at clinical efficacy and testing whether this is actually an approach to really treat Alzheimer's patients in the future.
Thank you, Frank. And let's return to the development wheel. Again, a reminder of the research underway. I mean, you'll be maybe overwhelmed by the amount, and it's actually true. It is happening, and it is because everything that Hendrik said is about the biomarker, the developments, the better diagnosis, the better way of prognosis, the better following the treatment over time, doing the natural course, using all these biomarkers. But there is so many things that are ongoing, and all the other mechanisms that we highlighted today are being studied. So inflammation, oxidative stress, the metabolism, omega-3, even metformin, the studies are ongoing. Masitinib, to name just a few, it's all in there. And if you take a good and close look at this figure, which is yearly updated, by the way, you can get a sense of what's happening. So, how is the future? I mean, we as old guys, we can say we have seen it all, but can we also look into the future? Hendrik, what's happening in the future?
Yeah, I feel extremely positive, and I also, and that is sort of, in general, I think, at least we can envision a future now when people who perhaps know that they have Alzheimer's in the family or so could actually be a little bit more active in regards to detecting onset of the disease in themselves. And that wouldn't be a bad thing, that you actually could But now I'm talking about the future. Since it looks like some of the anti-amyloid treatments actually do something important, and we have a lot of other things happening here in regards to other disease mechanisms. If one could then think about monitoring onset of pathology before it has happened, using biomarkers in blood, accessible, one could do it as a part of a health checkup in the primary care physician's office.
You think that's feasible to have it as health check in the primary care?
Yeah, but one has to be careful because there can't be screening at the moment. No. And it's but if one then do this monitoring and the blood biomarker starts to become a little bit abnormal, this could be a positive thing to know, and you could get evaluated for onset
to get
of
at least
pathology.
evaluated and perhaps even started therapy. And if you look at the future of therapy, we come from nothing. Now we have the monoclonals. Where do we end? What is the next step? I mean, will it be one treatment or will
no? I think there will be many treatments, and I think also that it's so nice to see this wheel of fortune, as you call it. But if then this personalized approach, what is the dominating pathophysiological process?
That's the word.
And then intervene there, and perhaps add on if there are other processes that are ongoing. And imagine how fun it will be to be a doctor in such a setting and dissect this together with the patient.
Yes, exactly.
And then perhaps initiate a treatment where you intervene and follow the effect with the biomarker.
And then start something else.
Exactly. And you invite the patient back and you said, this didn't work.
It becomes a really different sort of... We used to say to Alzheimer patients, well, you have the diagnosis, we can't do anything, and we'll check you up in 6 months. Now, it's a real proactive state, but you have to do something. You dissect the brain in the different pathologies, and you treat one by the other, and it will be a completely different attitude.
Yeah, I think we are entering a new era of Alzheimer's disease with more and more biomarkers, which puts us in the position to more and more understand the particular pattern of pathology in an individual, amyloid tau, and also aspects of neuroinflammation. And in the future, I hope that we will have a more targeted, more personalized treatment approach looking at these individual components. And the role of depression inflammation here, I think, very important because we can manage it, we can reduce it, and hopefully this is of benefit for the patient.
Thank you, Frank.
And are you also using lifestyle intervention and
yes, of course, using personally myself, yes, but we also tell it to the patients, and because they always ask, doctor, what can I do myself? What have I done wrong in the past? That's always difficult, of course, but what can I do in the future? And sometimes it's just your blood pressure, your cholesterol, living a bit more healthy and doing some more exercise can already help. We don't know yet, but
yeah, I think we know today that there are many lifestyle factors which have an impact on the risk and probably also on the course of dementia. And for me as a psychiatrist, I would like to mention one more, which is often in a way neglected, and that is depression. We know that depression is a risk factor for Alzheimer's disease, that depression accelerates the course of Alzheimer's disease. Depression is treatable. So my piece here is to consider depression and look for depression in individuals.
Thank you very much. It becomes a really personalized approach, and that's what I like about it as well. Yeah. So I think we've been looking to the past, in the present, and the future, Hendrik. Thank you for this great discussion. I enjoyed it, and I hope the audience did as well. And thank you all for participating in this activity. Please continue on to answer the questions that follow and complete the evaluation, please. It will help us to develop the program further for the next time.
This program was presented by Medscape Education Global.